All right, everybody, welcome back. We are continuing on, and in this section, we're going to really focus in on the different structures and organelles that make up eukaryotic cells. And specifically, we're going to be using this information not really just to sort of memorize, okay, this is this organelle and it performs this function, but rather how they symbiotically work together to produce different processes in the cell. Because that's really where we're headed, is understanding the different processes and the structural chemistry that's happening within the cell. So previously, we talked about how eukaryotic cells are different in several main ways, and critically that they resist the effects of antibiotics, which is what we discussed last time when we were looking at those differences in the ribosomes. Um, and one of the key ways that they resist effect is just the fact that they have these different organelles, and each of them performs a specific function, and that's what we're going to be sort of going through in this section. So again, you've probably all had this material before in other introductory biology classes or in your high school biology classes, where we're just looking at differences between plant and animal cells uh, and what organelles exist within them. So we're not going to spend too much time on this because this is most likely review for most of you. But just know, that there are specific differences, um, and we went through some of these even in our last section exam. And what I want you to focus on more is how these different organelles interact with one another and why they're important to the cell structure. So we're going to start with the nucleus. Um, So the nucleus is really the defining characteristic of eukaryotic cells. So rather than having free-floating DNA as we saw in prokaryotic cells, we're now sort of confining that DNA to a specific area, and this area with its own phospholipid bilayer, creating a membrane around this again. And this is really where the genetic material for the cell and everything that's going to encode for all the different types of proteins and all the different types of syntheses that the cell is going to make are all enclosed in this nucleus. And like I said, this cell membrane in this case is called the nuclear envelope. The next organelle is probably, as you all remember from your high school biology, it's the powerhouse of the cell, the mitochondria. Um, and really, a few things that really make this organelle special is that it has these two membranes which surround it. One of them is highly folded, and the other one um, is sort of keeps everything trapped in this useful form. And you can see that on the inside, you have all these ribosomes. So again, this is where energy um, can be derived from food for the cell. The next organelle that I want to talk about is the endoplasmic reticulum. Now, there are two types of endoplasmic reticulum. There's the rough and the smooth. And the rough contain again these ribosomes, which again are going to be important for encoding new proteins. The next organelle is the Golgi apparatus. So again, we have a very similar structure to the endoplasmic reticulum, but the process here is really to package proteins that are produced in the rough endoplasmic reticulum. So this is where things are sort of processed so that the cell can make use of already synthesized material. And so let's start to get a picture of what synthesis starts to look like in the cell. So first we have um, the nucleus, which provides the instructions for protein production. Then we have proteins that are made in the endoplasmic reticulum. So the ribosomes are assisting in creating these proteins. And then these proteins are packaged into the vesicles to the Golgi apparatus. And then from there, the proteins receive any final modifications they have in the Golgi apparatus, and they're able to be secreted from the cell or go to various locations within the cell. And this is really how we start to think about where chemical processes are occurring in the cell and what steps are occurring so that a cell can create energy or synthesize products. So we talked a little bit about energy synthesis in the cell, but the other part that we want to talk about is sort of recycling and digestive aspects of the cell. And these are primarily produced by the lysosome. So essentially, they're recycling centers where from that Golgi apparatus, a lysosome is able to lyse to eat specific things for digestion and any other damaged parts of the cell. They're also able to take food particles and other parts out of the cell. So again, essentially, it's a recycling center with these digestive enzymes that really break down worn out cell parts or molecules so they can be used to build new and other structures. And probably one of the most sort of overlooked aspects of the cell is the cytoskeleton. So this is really the meshwork of proteins and fibers that carry out a variety of functions that include sort of the cell transport that we discussed previously, cell movement, and movement of structures within the cell. And there are several different types of filaments that contribute to this. There's microfilaments, intermediate filaments, and microtubules. And what this really is, it's a network of protein fibers, and they assist in cell movement, cell transport. So you can think of flagella. These are the structures that really dictate how a cell is going to move. And then lastly, I want to talk specifically about chloroplasts. So we discussed this briefly in our plant lecture as just sort of a unique structure to plants and algae cells. But this is it's an interesting organelle in that it has two membranes and it has sort of this internal stacked structure of discs. And this is where the site of photosynthesis happens. Occurs in these regions. They're able to capture and convert sunlight energy to some sort of usable energy form that the cell can then use. And we also talked briefly about the endosymbiosis theory, that essentially the way that eukaryotic cells came to be is that free-living prokaryotic cells were able to engulf other free-living prokaryotic cells, forming these new eukaryotic organelles. So essentially these organelles, such as chloroplasts, were once prokaryotic cells on their own, potentially a cyanobacteria, as we discussed previously. They were able to eat that and create these new free-living cells, and they basically take the energy and hijack what that old previous prokaryotic cells and put it to work in a new cell. And there are a variety of different organelles that's been hypothesized came into being this way, namely mitochondria and chloroplasts. So in summary for this section, all living organisms are made of cells, and new cells arise from existing cells. And that's really the convention of cell theory biology. Next, all cells are enclosed by a cell membrane, which controls the passage of molecules between the exterior cell and the cytoplasm. And we discussed various ways that they can move through these membranes through simple or facilitated diffusion or by active transport. We discussed that when we're talking specifically about water, water crosses cell membranes by the process called osmosis. We also talked about ribosomes, how the complex of RNA and proteins synthesize new proteins. And then we also discussed the specific organelles that are characterized in eukaryotic cells and the differences that occur between them. So really what this section is getting at is we're starting to understand the structure of the cell, how those structures interact, and what we're going to be moving towards is understanding how these cells work together to create digestion, metabolism, and understanding these higher levels. So hope to get to you next week where we're going to be covering those topics. All right, get up, take a stretch, go outside, take a walk if you can, and then come back for next week. All right, thanks, Paul. Bye.